In this lecture, we'll talk about how we go about mapping mental functions onto the brain. So one way that the goal of cognitive neuroscience is sometimes framed is as linking molecules to mind. The idea is that when we engage in cognitive functions, there's these sort of mental processes that we can't see that are happening in our head. Those ultimately rely on the activity of neurons in our brain, which we can sometimes measure. And then ultimately those bottom out in sort of, biological, metabolic, molecular processes happening in our brains. So one way to think about the problem of mapping minds to molecules is, on the one hand, we have these mental functions that we're interested in, but we can't see. So things that psychologists talk about like memory retrieval. The only way we can try to get a handle on those things is by having people perform psychological tasks and observing their behavior on those tasks. So for example, I might show individuals a set of pictures and then give them a task where they are shown individual pictures and have to say, is that picture old or new? Did you see it before or not? And then what we can do is indirectly observe brain activity while they're performing that task. We see their behavior directly. We see their brain function indirectly using tools like functional MRI. Now, the design of an fMRI study is challenging for a number of reasons. One of them is that the magnitude of the fMRI signal on its own is not meaningful. It's not as we're not measuring a quantitative signal that has a particular unit. We're basically measuring kind of an arbitrary signal. So the only way we can look for activity is to compare signal between different conditions and look for relative differences. So let's say that we wanted to do a study to understand the brain systems that are associated with the perception of food. Now, one of the most common approaches to performing fMRI studies is known as the subtraction logic. This was a logic that was originally described by Donders, the experimental psychologist, in the 19th century for the analysis of reaction times. The idea is that we acquire data under at least two conditions, and those conditions putatively differ only in the cognitive process of interest. So the idea is that we collect brain images acquired during each of those conditions, as shown in the figure to the right from the early Peterson PET study. One of those parts of the data is collected while the person is being stimulated, another while they're doing some sort of control condition. And we look at the difference between the activity under those two conditions. And what we have to do is infer that that activity is related to whatever process of interest has been subtracted out. Whatever psychological processes differ between those two conditions, those are thought to be what's driving that difference in activity. So let's say for our food study, we might show people a block of pictures of food items for, say, 24 seconds. Then they rest for a moment, and then we show them 24 seconds of non-food images. Let's say we decide to use office supplies. Then we show them food images again, we show them office supplies again, and we do this enough times to collect enough data to be able to estimate the brain's response to these different pictures. So what does that look like? Well, first let's ask, what would the neural response look like if I show a person food pictures for 24 seconds and there's a brain system that only responds to food images? Then we would expect its activity to sort of go up, as I'm showing here in this sort of this boxcar response. So as soon as the food pictures start, activity goes up. As soon as the pictures go away, it goes down. Now, let's say that there was a brain region that was only responsive to non-food images. That's going to be shown here in blue. Well, as soon as we start showing the office supplies, its activity is going to go up, and it'll stay up until they're gone, and then it'll go away. And if we do this repeatedly, we would see that the food area would go up whenever there are food pictures on the screen. The non-food area would go up whenever there are non-food pictures on the screen. Now, this is, remember, the neuronal response. This is the firing of neurons. But what we know about the fMRI response is that it's relatively sluggish with respect to the blood flow response. Sorry, with respect to the neural response. So in this example from Randy Buckner, we see that the response starts about 1 to 2 seconds after the neural activity has started. And then the blood flow response peaks around 5 or 6 seconds. And in order to find the areas in the brain that are showing the expected response, we need to take this sluggishness into account. Now, one really fortunate thing that was established in the 1990s is that this response is at least roughly linear. That is, that it's basically the response to any particular set of events is just a linear combination of the response to the individual events. And that makes building our statistical models much easier. All we need to do is take those hypothesized neural responses, which are the square waves here, and do what we call convolving them with our expected hemodynamic response. And that gives us our kind of hypothetical blood flow response, which is what's shown here in the solid green and solid blue lines, again, for the regions that respond either to only food items or only non-food items. Now we can look in the brain and ask, where in the brain does the blood flow response, the fMRI signal, follow those particular functions? And here's an example using data from a study by Paul Smeets and colleagues that shows an actual voxel in the brain that follows that particular pattern of only responding more to food images than to non-food images. So you can see that the sort of thin line shows the actual data from this one location in the brain, in the orbitofrontal cortex, that has activity that's significantly higher for food versus non-food images. Now you see that there's a lot of noise. It's a very jiggly line, right? But nonetheless, you can see that it goes up roughly when the green section is going up, where there's food objects on the screen, and it goes down when there's non-food images on the screen. So the question is, what do we conclude from this? The data show us that orbitofrontal cortex activity is significantly greater for this set of food pictures than non-food pictures, but we want to know, what does that reflect in terms of cognitive functions? And in reality, that difference reflects whatever differences there are between those two conditions. Now, obviously, we know that the presence or absence of food stimuli is one of the things that differ, but we always have to keep in mind that there could be numerous other potential confounds that could drive that difference. So, for example, the person might be more familiar with food pictures than with office supplies. The office supplies might have different visual features, different colors, or different spatial frequencies in the image. Or it might be that this is a region that just becomes active whenever this person experiences something that's arousing, right? And it might not be specific for food. It might be related to anything that's arousing. It's also important to keep in mind that before we can ever do any statistical analysis on the functional MRI data, we have to do a lot of what we call pre-processing to get the data ready for statistical analysis. This is just one example from a tool that we've generated called fMRI Prep that does pre-processing for fMRI. And you can see all the different stages that are involved. And this leads to a lot of potential for analytic flexibility. We estimated in a paper that just for a single software package, there were almost 70,000 possible workflows that one might use that were all, none of them were sort of unreasonable workflows. And we know now that these differences in data analysis can have major effects on the results of a statistical analysis. This is a paper we published in 2020 where we collected a data set of individuals performing a task, an fMRI data set, and then we gave the data out to a number of different analysis teams. We ended up getting results back from 70 teams, each of which analyzed and answered a number of different hypotheses about whether there was activity in a particular part of the brain for a particular aspect of the task. Now, if everybody agreed, then all of the numbers in this plot here would be either 0 or 1. This is a plot showing the proportion of teams that reported a positive effect for each of those hypotheses. In fact, there was a ton of variability. For 3 of the hypotheses, the ones on the left, most teams agreed that there was nothing going on. For one of them, on the right, about 85% of the teams agreed that there was significant activation. But then for 5 of the 9, 20 to 40% of the teams said that there was significant activation. And if we look across the teams, there were 33 different patterns of outcomes for the 70 teams. So this is a striking and worrisome amount of variability in the answers that one can get from the same data given differences in analysis pathways. And it really highlights the need for any researcher to run the data through multiple pathways and make sure that the results are robust to any specific features of the analysis. Now, moving from the sort of brain side to the cognitive side, let's now ask, what is it that we're actually mapping onto the brain? So, in general, when we do an experiment, we look at the things that psychologists talk about, mental functions, things like working memory or cognitive control or goal selection. And we refer to the framework that sort of describes those functions and their relationships as our cognitive ontology. What are the parts of the mind? Right. So if we think about what are we trying to do, right? We're trying to understand how particular cognitive functions relate to particular brain systems. We can't see those cognitive functions. The only way we can see them is using cognitive tasks. And so it becomes essential to understand what is it that any particular cognitive task is doing in terms of the psychological functions that are being engaged. So let's look at an example that shows us that cognitive tasks are rarely pure measures of any particular psychological process. So as an example, let's use what's called an in-back task. This is a task that's thought to reflect working memory. If you do a PubMed search for, in quotes, in-back working memory task, that means people are using exactly that phrase. You can find 230 papers that use that phrase. It's a task in which the person is presented with a stream of stimuli, and they have to say for each stimulus, is it the same as the one that was presented in stimuli back, right? So for example, a 1 back, they would say, is it the same as the previous stimulus? A 4 back task would be one where they have to say, is it the same as the stimulus you saw 4 stimuli back? Now, this is thought to reflect working memory, but it turns out that it may not actually reflect working memory at all, at least working memory as it's reflected in other types of more accepted measures of working memory, things like digit span tasks. So we now know that in-back performance actually doesn't correlate across people with these other accepted measures of working memory tasks. On the other hand, it does correlate with other factors beyond working memory, things like processing speed. And thus, we can't simply interpret the differences between in-back conditions in terms of the construct of working memory. We have to understand all of the possible psychological processes that might be involved in that particular task. So in summary, we can use experimental logic borrowed from cognitive psychology to infer that particular brain systems are associated with particular mental processes. But we have to be careful in establishing those mappings between brain systems and mental functions.